Hello, everyone. This is also my first time sat, so I'm excited to be here today. I only been in Monell in like 3 months, so this is all very new to me, but it's very exciting. My name is Gabrielle. What we will be talking today, it's about the microbiota in the brain. So before we start, just to give you a little bit. Oh no, this is not working. So just to give you a little bit of an overview of what I'll be talking today. So I've been asked to give you a little bit of a brief description of my journey as a scientist and how I got here and how I got into this world of the microbiome. Then we will talk a little bit about what is the microbiome, but also about the gut-brain axis, so how then the microbiome can actually get to the brain. We will talk a little bit about some papers and what is the evidence out there for the microbiota to be changing brain function and behavior. And we will end with maybe a little bit of an overview about the challenges of the field and what does it mean for health in general. So to start off, as you can tell by my accent, I'm not from here. I'm from Brazil. I'm from this town called Belo Horizonte. It's a kind of a big city. I did my undergrad there. And once I left high school, I didn't really know what I wanted to be because in Brazil, when you like biology, they tell you to go do medicine. And I did not want to do medicine because I don't like to work with people directly. I don't like to be responsible for people. So I went to biology. And to the very big disappointing of my family, who thought I was going to become a high school teacher, but here I am. So during my undergrad, I found out about research. I started having research experience. This culminated with my bachelor thesis being on the role of the vagus nerve in maintaining energy metabolism in the liver. And that got me really excited about science. So once I had that experience, I wanted more. So then what I did is I applied for a bunch of internships to try to get a little bit more of a research experience to know if that's what I really wanted to do. So that's what brought me to Caltech. I applied and I got a summer undergraduate research fellowship, and where I had my first experience working with the microbiome. Here I worked with the role of the microbiome in Parkinson's disease. It was a really good experience for me. It was my first experience outside Brazil. So that got me really excited. I went back home and I immediately started to apply for different PhDs. And then I traveled across the ocean again and I went to Ireland. So I ended up in Cork, which is a city on the south of Ireland. And I worked in this place called the APC Microbiome Ireland, which is this big institute with people from different fields, microbiology, neuroscience, gastroenterology, all investigating the microbiome and the impact of the microbiome on our health. So there I did my PhD in neuroscience. I was working in understanding how the gut microbiota can influence the way we respond to stress across the day. So after that very good experience on my PhD as well, I continued wanting it more and I continued wanting to work in science, and that brought me to Monell. So I am here. I'm working now, continue working with the microbiome, continuing this field. I like collecting mouse poop for a living. So I'm working on trying to understand how the microbiome changes the way we eat, and more specifically, how the microbiome helps us decide when we eat and how we eat. So yeah, that's about me. That's how I got here. And but moving on about the main topic of today, which is the microbiome. So the microbiome is very big. It has we have over 100 trillion of microbes on us. More specifically, when we talk about the gut microbiome, it's important to know the amount of microorganisms that reside in our gut. It's estimated that about 10 billion bacterial cells per gram of feces. And when you think about you as a whole, it's estimated that about half of your body weight is composed of microbes. And that value will kind of vary across the day. So like after you go to the bathroom, you kind of become more human and less micro because you lose some of those grams. But overall, we have sort of this ratio of 1-to-1 between human cells and microbe cells. But the microbiome is essential for health, and that's very evident when we think about digestion, because microbes participate in our digestion process. It digests things that we as humans don't have the ability to. It digests complex carbohydrates. It helps us digest fiber and produces a bunch of nutrients for us that help us become healthier. Also, the microbiome is very important for our immune system and for our resilience to the environment. This is, I think, is a very nice example that actually the microbiome helps us prevent infections by pathogenic bacteria. So, for example, one very common treatment today in terms of health is fecal microbiota transplant, where we transplant the microbiome from one human to the other. This is used because we have a bacteria in our gut called C. diff, so Clostridium difficile. And this bacteria, when it starts to proliferate too much, it can become pathogenic and cause a bunch of gastrointestinal symptoms. But when you get a donation from a human donor and you give the microbiome to somebody who is infected with C. diff, the person actually becomes healthy again. So this is already showing that the microbiome is important for our health, and it can actually be used as a target to improve overall health and fight infections. Although we know that the microbiome is important for these things like metabolism and food and nutrients and infections and immune system, the idea that the microbiome could actually play a role in upstream, like the brain, it's kind of older than a lot of people thought. So there is this guy that published these works around the early 19th century called Mechnikov. This guy, he believed he was called the father of innate immunity. So he won actually a Nobel Prize for his work in immunity. And he believed that microorganisms play a big role in our health. Actually, he believed that consuming lactic acid bacteria could actually help prolong life. This work has culminated in this publication from early 1900s called The Prolongation of Life. And actually in this study, what he did, he's investigated these like very old people in Bulgaria, like over 100 years. And he was trying to correlate how they were getting older with the fact that they consumed a lot of yogurt. So this was one of the first ideas that the microbes that were new actually helped us stay young, and some sort of idea that may actually have beneficial effects for your brain. Actually, to today in South Korea, there is this yogurt with his face on it, which I think it's very interesting. So these were the first ideas that in terms of like science, that the microbiome could be important for brain and aging. But our culture itself is already embedded with references to the gut microbiome. So we use sayings daily like trust your gut feeling, gut wrench. So we already know that somehow the way we feel is connected with our overall health, with our well-being. So would there be any evidence scientifically that these microbes or whatever is going on in your gut can actually impact brain health? And this is where we come today to the field, what we call the gut-brain axis. So the gut-brain axis, the interaction between the gut and the brain, more specifically, is the bidirectional communication between these two organs. So whatever signals are being sent from bottom up and from up to down, and how these are overall managed to drive health and to maintain health. So when we think about the gut-brain axis, right, we are thinking about this specifically. So we have the brain, we have our gut, we have our microbes here, but we have a bunch of other components in between. So how these two systems communicate is first by neuronal connections. So you have, for example, the vagus nerve with a direct connection from the gut all the way to the brain. You have hormones that go into the circulation, both produced in the gut and derived from the brain, that can actually affect how each other work. You also have our microbes producing microbial molecules that can actually arrive in the circulation and then directly affect the brain. And you also have immune signals. So the gut is full of immune cells, and these immune cells also produce these infractors that can shape our microbiome and shape our brain. So it's this bi-directional communication. Things affect each other up and down. So um, then becomes the big question, how can, right? And in science, when we want to understand how something affects the other, the first thing we do is we remove that variable and we test it out to see what happens. So what if we got a human and we remove the microbiome? What happens? Well, there is actually an example of that. It's this boy called David Vedder in the 70s. He's called the bubble boy. He had a very rare immune disease and he was actually removed from the uterus of the mother and he was put in this bubble. And then they had to like sterilize everything that came in and out. So he was maintained 100% sterile. Actually, NASA developed him a little astronaut suit so he can actually walk around and explore the world. So this goes to show that it's not practical. Unfortunately, we cannot put a human and take out the microbiome to investigate this. So when we can't do something with a human, we do it with a mouse. So scientists have been doing this since the late 70s, where you grow an animal in a sterile environment. So the same way that that boy grew up, these animals grew up. They grew up completely sterile. Everything you put in, food, water, bedding, everything you put in that cage has to be completely deprived by the microbes. And they grow like that. So what scientists find out is that when they test these mice, they behave differently, right? So if you put the mouse in an arena without anything, he will prefer to go to the corners. He will prefer to go to the places where he feels safe. But if you put a germ-free animal in the same arena, he will go more to the center. He will explore more. He shows less anxiety-like behavior. So this was one of the first reports that microbes could actually be playing and shaping the way we behave. So what was interesting as well is that when researchers analyze the brains of these animals that behave like sentries, they actually have different patterns of expression in the brain regions that encode for this behavior. So that's when the early 2000s, 2010s and late 2000s was when we got the first big reports that the microbiome was shaping brain. So what I would do now is that I will go through a few examples and a few papers that I think are interesting and show the evidence of the microbiome on the brain across the lifespan. So we start in early life all the way to aging. So I ask you, please stop me if you have any questions. Don't be afraid to ask anything. This doesn't supposed to be just talking at you all the time. So feel free to ask me anything in between if something is not clear. So we will start at birth, right? So when we are born, we have our first contact with the microbiome because inside uterus, we are completely sterile. There is no microbes when you're inside your mother. But once you come out, that's your first contact with the microbiome, and that's what we call the seeding of the microbiome. And the way you are delivered influences how your microbiome will look like for the first months of life. So when a baby is born by natural birth, so it goes to the vaginal canal, you can see that it doesn't matter from the very beginning. So for 4 days of age, all the way to infancy, their microbiome already looks very well established. But when a baby is born by C-section, you can see that we bypass that initial colonization. That happens because the microbiome and the microbes that are around the vagina and the anus, they actually seed the microbiome of the baby. So what happens is in the first days of life, your microbiome looks very different than somebody who was born naturally. So researchers then wanted to understand if a germ-free mouse has a different behavior, if we know that the microbiome affects the way we behave, if you have a different microbiome in early life, will that shape the way you behave later in life? And that's what they did. So they run this test where we put a mouse and we have two other mouse in these chambers, right? So the mouse number one here is a mouse that they already know. It's his friend, he's familiar with. And the other side, there is a novel mouse, a mouse that is different, that he never had contact before. Generally, mouse are sociable animals. So what they would do is they will go interact with a novel mouse, something that is new to him, and he will explore this new mouse. And what we found is that mouse, when they are born by vaginal birth, when they're born by vaginal birth, they show this preference for the novel mouse, as you would expect. They are more social. But when the mice are born by C-section, they lose this preference, meaning that the social behavior is being altered later in life just by having a different microbiome early in life. And what researchers did then was to characterize the microbiome between these two groups and see what was missing. And if we give back to the animals what was missing in the beginning, we show that we can recover the sociability. So here, the vaginal born, again, showing the social preference, it lost when the animals go to C-section. But if we give the microbes that were missing back, they recover that same behavior. So continuing early life, bring another example of maybe the interaction between diet and the microbiome. So when you give mice a hyper diet, they become obese, and that causes an array of disruptions to the body. And one of the disruptions that eating a diet enriched in fat is that you change your microbiome. So researchers, what they did then was to give a control diet to some mice and a diet rich in fat to other mice. And what they saw is that they tested the offspring of these mice. So to see how the impact of giving the mothers a diet rich in fat would affect their babies. And since I showed you before that this seeding of the microbiome, the passage of the microbiome from the mother to the baby is so important to look at social behavior, this is exactly what they looked. They did the same very test that I explained to you before. The mice, they have the chance to explore a chamber with a novel mouse or with a familiar mouse. They tend to prefer the novel one that is new to them. And here you can see that the animals that receive the mothers receive the control diet, they have normal behavior, they interact more with these novel mice. But when the mothers are given a half a diet, this preference is lost again. So again, this shows that the diet here being an important factor, but how then could you pinpoint this to the microbiome? To do this, they decided to, again, analyze the microbiome and compare the difference between the microbiome of the control mothers to the high-fat mothers and understand what changed. And they decided to replenish the offspring with a bacteria that was missing in the high-fat diet mothers. And what they saw is that by doing that, they can again recover the same normal behavior later in life. And this was more evidence that in early life, when you change the microbiome, either be by maternal diet, be by mode of birth, and I can cite other factors that change your microbiome, you're actually changing some behavioral aspects later in life. Now we move on to adulthood, right? In adulthood, one really cool experiment for me that really, I think it was really cool in early 2000s was this paper that basically what they did is that they get a mouse and they restrain their movements for, I think it was 30 minutes. And they measure a bunch of stress hormones that are released upon the stress, right? And what they saw is that when you have a germ-free mouse, again, without any microbiome, these animals have this big release of stress hormones that the other animals don't have. So this was the first report that the microbiome can shape the stress response, so the way that we respond to stressful situations. So after doing this, some researchers started to question themselves if the microbes could have any relevance for mental health conditions, as depression, anxiety, and other conditions are often called stress-related disorders because their beginning has a lot to do with stress. So what researchers did is that they got the microbiome from people that were diagnosed with depression, and they gave that to germ-free mouse, and they saw how these mice behave. And what they saw is that these mice started to showing some depressive-like phenotypes. I mean, here, unfortunately, you cannot ask a mouse how they are feeling, and they will not respond a questionnaire to tell you that they are feeling depressed, but we can run a few tests to show that these mice, they move less, that they explore less, and these together, these findings show that they have somewhat of a more depressive-like phenotype. On the other hand, another example for the implications for mental health is in the case of social anxiety disorder. researchers again took the microbiome from humans, from patients that were diagnosed with social anxiety disorder, and they gave that to mouse. And then the mouse start to interact less with other mice, meaning that somehow the microbiome is conveying signals that are important to maintaining normal mental health. Continuing adulthood, I talked to you about mental health, about social behavior, but metabolism is very important, and the way we eat is very important. And one cool thing about animals without a microbiome is that they don't get fat when you give them Western diet. So here what researchers say Western diet is diet rich in processed foods. So it's basically our diet. So diets that are rich in sugar, rich in fat. So when you give that to a germ food, so then we give the obese mouse microbiome to a germ-free mouse. And then the germ-free mouse then became obese, meaning that these obesity-drive signals was somehow being conveyed by these microbes that were inside their guts. This might be explained somewhat by the way these animals eat. So, for example, if you take out the microbiome with antibiotics, you can see the animals generally tend to eat more sugar pellets. So that increases that preference for sugar. So somehow these microbiome alterations that we go to makes us, crave more of something. It somehow relays some sort of need that drives obesity and dive metabolic disorders too. So if we can transfer bad things, so if we can transfer depressive phenotypes and less social phenotypes, why couldn't we transfer good things? So what if we put the microbiota of a young mouse on an old mouse? What does it happen then? And this was exactly what some papers try to do. They transplant the microbiome from aged and young mice into other mice and see what happens. And one thing that happens as we grow older is that our memory starts to get worse and worse. So what researchers were trying to do is try to measure the memory of these mice and see if giving them microbiota for younger mice could actually help prevent some of these aging effects. So this test is called Morris-Water maze, and basically what you do is you put a mouse in a big pool of water, and they have like this tiny little platform that they have to find and they have to swim to. So they so in the beginning, when you put them for the first time, they will go around and around and around looking for the platform, looking for the platform. The more you do it, they start to learn where this platform is. So they start to get there faster and faster. So the more trials you do, the faster the mouse you get at it. And if they have cognitive decline or some sort of memory impairment, they should find it harder to find this platform. So when they gave FMT to an aged mouse, FMT from older mouse, the memory actually got worse than they would from a younger mouse. And you can see here that the mouse that received the FMT from older, they start to get, they don't perform as well as the animals that received from younger, meaning that somehow these microbiota convey signals are actually modulating memory procedures as well. And to kind of wrap up this part, another aging example is the example of neurodegenerative disorders, where when you have a mouse with a control mouse with complex microbiota and you have a germ-free mouse without anything, you go and you give them Parkinson's. The germ-free animal actually show less motor dysfunction than the animals with microbiome, showing that somehow not having a microbiome is somehow protecting them from this Parkinson's phenotype. And when researchers then gave Parkinson's microbiota to mice, they saw something similar. So the animals that received the microbiome from healthy controls versus the animals that received microbiota from Parkinson's patients, the ones that received from Parkinson's patients had worse motor symptoms than the one from healthy controls. So that meant that somehow the microbiome has some sort of role in the progression of the Parkinson's disease, in the progression of aging. So this was just like a brief overview across the lifespan of key evidence points that the microbiome can change the brain. But I think a question that we have to ask ourselves when we are in science is why? Why does this happen? What is the benefit for us to respond to microbial signals? Why do that we do it in the first place? And something that a lot of people forget is that microbes have been here way before us. And microbes are way older than humans, and we evolved in a planet that was full of microbes. We co-evolved with these microbes, and we generated kind of this symbiotic collaboration with these microbes that in the end, the microbes are part of our environment. And in order to survive, in order to adapt, evolutionary speaking, you have to learn what is going on around you. And this is what microbes do. The microbes are the interface between you and the outside, be it in your skin, in your mouth, and in your gut. So they are part of the transmission of the signals of what's happening around you with what is happening inside you. And that helps us adapt, that helps us survive, and that helps us respond to the environment. And that might be a good thing, because in the end, it means that if you target them, maybe you can get better. And this is even exemplified in research that there is plenty of gut-brain axis research and microbiome research that has been using different animal models, from less complex models such as worm in C. elegans, then you go into flies like the Drosophila, you go to zebrafish, to mouse, to humans. All of these models have shown some sort of gut-brain interaction of some sort. So it helps us understand that throughout evolution, these microbes may have played a big role in our health, and they continue to do so. And I think that's why a lot of people get excited about the microbiome. And whenever a paper comes out, you will see in the news, you will see it in the newspapers, all about these big claims that microbiome is going to change the world, and how your gut health is important and you should take care of how you eat. And, yes. I think I showed you enough reasoning to believe that the microbes have some sort of effect on the brain, but there is still a lot of research to be done behind this news. So one of the reasons why a lot of people, and in general, a lot of the research field got really excited about the discoveries that the microbiome changes so much, comes to the fact that we started to sequencing those microbes. We started to understand how the microbial composition of us look like, and we started to notice that in a bunch of conditions, the microbes correlate with disease. So patients with Parkinson's have a specific microbiome that is different than other aging people. People with Alzheimer's is the same. People with depression is the same. So people started really getting excited about the fact that you have this different microbiota composition in people with disease. So it becomes, can we intervene in disease using the microbiome? And it's cheap. I mean, it's microbes, so you can grow them pretty cheap. So people get excited because it's new, it's cheap therapies. It's non-invasive. Okay, people are talking about fecal microbiota transplants in humans, and that doesn't sound super nice, but still it's non-invasive. And it could be used to treat a large spectrum of conditions. If it is really true that microbiome correlates with all of these different conditions across the body, maybe we can treat almost everything. But that's when it becomes confusion, right? Because correlation is not causation, right? So just because things correlate with each other, it doesn't mean they are true. So that's by becoming tangled in that sheet, and the per cheese per capita consumption correlate with each other. But I don't think the amount of cheese you consume implies your likelihood of getting tangled in your bed sheet at night. So these two things don't necessarily match up. And this is where the field is moving forward. It's moving forward to that initial findings that I told you about, somewhat tiny experiments of like giving the microbiome, sequencing the microbiome to trying to understand which are microbes doing it, which are the molecules doing it. So first, we run sort of these microbiome associations. Does this person with Alzheimer's have a different microbiome than my controls? And then once I find that, I go maybe to a mouse model or a fish model, and I investigate if taking the microbiome out by antibiotics, by a germ-free, do I improve those conditions? Then I maybe transfer the microbiome until I get to a point that I can pinpoint those microbial effects to single bacterial strains or single molecules or single metabolites. And this is where the health benefit will be later, where we identify these specific maybe strains, microbes that are driving all of this. But there is still also some important confounds to pay attention to, and one of them is geography, because the microbiome is shaped by what we eat, and the diets vary hugely across the globe. So there is, I think, this very cool study that took microbiome donors from 3 different places. One was the US, one was from Fiji, and the other one from Guatemala, and they gave this microbiota and they colonized mice with it. And then they did a Clostridium infection, which is the same example that I used in the beginning, but with the mouse. And they saw that the mouse from Guatemala were the most resistant mouse. So there was something inherently about the Guatemala microbiome that made them resistant to infection. So they were like, can we harness this microbiome then to prevent mouse from getting sick with infection? And that's what they did. So they got the most successful mouse that was from Fiji, and they co-housed them with mouse from Guatemala. And because mouse are coprophagic, they will eat each other's poop, the microbiome become more and more similar with time. And then the mouse from Fiji start to become more resistant to the infection. So that goes to tell us that where you're from actually dictates what your microbiome look like. So that's important to pay attention of, because there is an overrepresentation in the research of microbiome from North America and from Europe. So that limits the knowledge we currently have about what a healthy microbiome look like. And these are also important to know, very heavily industrialized nations. So it's nations that we consume a lot of processed food. So our microbiome not even maybe the healthiest or the best to define as healthy. So there is a lot of work still to be done to identify what is a healthy microbiome and understanding how we can target it to promote health. So this is where I'm coming to an end, and I was way faster than I expected, but it's what is the future looks like, right? There is a lot of challenges still in the microbiome field. It's trying to understand what does a microbiome look like, trying to understand what are the effects of lifestyle on the microbiome. What can we use as a control? Do you use your mother or your sister, your parents as your control, or can I use any random person in the same city as you? Is these questions that scientists are trying to understand now to define where the field is going. So I personally think that there is a lot of potential here, but it still has a lot of work to be done. And I hope I convinced you that the microbiome somehow plays an important role in our health, also in the brain, and not only on other aspects. But just as a conclusion, so what we saw today is that the microbiome can influence brain function and behavior, but more research is needed to go beyond correlation. And lately, we finished with a kind of message of hope that maybe in the future we can see more and more microbiome-based therapies to help us be more healthy. So improve conditions like depression, like anxiety. And, those are going to be really helpful, because in countries where access to medication is very difficult, if you have a microbiome-based intervention, that might be really easy, might be much helpful. So we still have a long way to go, but I hope I got you excited for the field. And, if you have any questions, please let me know.
You showed us that the microbiota seems to be involved in a good way, in a bad way, for depression, aging, memory, metabolism, just it seems to do everything. How do you think you can harness that in a more targeted, specific manner? You don't want to try to treat Parkinson's and make somebody obese, for example. So how do you see the future for that?
Well, I think that's a challenge, right? Because I think what people are talking about in moving towards is more personalized medicine, is trying to understand what your microbiome look like and trying to design a plan for you instead of trying to generalize something that will work for everybody. And also in conditions, what researchers are trying to propose is the microbiota as a target to improve maybe also therapy response. So like in the case of Parkinson's, can we improve how well the person responds to the medication, to the treatment by targeting the microbiome?
But how can we make sure that like we change the microbiome of the individual, either through FMT or through more targeted approaches, that we're not having off-target effects that cause diseases that we really don't want to be causing?
I don't think that we know that and the answer to that for certain yet. I think because we haven't classified what does good and what does bad. So we would try to increase something that we think is good and that might cause some upsets in other areas. But
so how do we move forward? Do we have to take every single bacteria, figure out what every single bacteria does? And you said there's billions.
Yeah, so I think that what people are trying to move to is trying to understand the community as a whole. And the thing is, generally, if you have what people define as healthy bacteria, if you have that in a level that is good, you're not going to cause any detrimental effects. So what people try to do and is that it is, for example, biobanks of bacteria that people can donate to today, that you have to be a certain BMI, you have to be a certain age, you have not to consume meat, you cannot smoke, you cannot drink. And like this is what people call optimal microbiome. So this is what people are going to try to strive to. And hopefully by focus on that, you can avoid off-target effects.
You showed that funnel that sort of ended with like strained molecules.
Yeah.
How many of these projects have actually spun it down to a level of understanding of molecules?
So I think with humans, not a lot. With mouse, there is some works. I know some with like social behavior that they were able to pinpoint it down to a single metabolite. Some works with a single strain, also with like cases of autism models, but they are not the majority. They are not a majority. So I think then it becomes also the fact is how do you translate that to humans? And I know some studies, especially social behavior, that are trying to give the same bacteria we give to the mouse to like children and see how they behave later. And those are still early reports.
So, like the microbiome of your skin is not the same as your gut, is it? Same with your nose, do they talk to each other? It is like this layer on.
Yeah, there's some sort of so like the microbiome generally of your skin would look very different than your gut, because in your gut you don't have oxygen. So like most of the microbes on the gut will be anaerobic, so they will not use oxygen. While in your skin, most of them will be. So those are very different microbiome. A lot of the microbiome in your skin will be important to prevent infection, to maintain this kind of layer that protects you from the outside. More of the microbiome in your gut, a lot of them will be focused on nutrition, on breaking down things that you cannot break down. So they have kind of like these different functions, and they will be adapted to the specific conditions of your body and the body part that they are.
Is it just your diet even that affects your microbiome?
A lot of things. Like that's the problem. Microbes seems to respond to almost everything. exercise would change your microbiome, stress would change your microbiome, diet would change your microbiome, age would change your microbiome, even the time of day would change your microbiome. So it's a very, very dynamic community. And this is also one of the challenges, right? Because if you go to the doctor and you get your microbiome sample to understand how you can target it to improve your health, 2 months later, when you go back with your results already, your microbiome might not be the same because you've been through, I don't know, you were giving antibiotics by your doctor and your microbiome now looks different. So it's how do we get around those things that will be key for the future. Yeah, I think IBS or irritable bowel syndrome is a good example that people, some cases have reported that people get this FMT and they get better because the microbiome gets better, but with time, these effects are going to be started to be washed out, and then these effects might disappear. So we might look into a future where you might have to get FMTs often. And some researchers are developing FMTs in pills. So a lot of FMTs early days were to enema. So you had literally to inject yourself with the fecal matter from somebody else. But nowadays, researchers are even investigating pills that are kind of like an FMT that you take, and they because of based on pH of your stomach and your gut, they will only release in the right place in your gut, and that you can replenish the microbiome like that. So it's not going to be as disgusting.
Curious from like a researcher or like a personal perspective,
Yeah.
what are your thoughts on over-the-counter prebiotic and probiotic supplements?
Complicated because a lot of companies will get there was it could be nature, a new big study was released showing that this strain does this. And then a company, because they don't need necessarily FDA approval to sell those things, they would try to back themselves with those studies, even though those researchers never intended to say that that bacteria may produce some sort of result in humans. So I think we have to be very careful. I would more intend to believe probiotics and prebiotics that are based on human trials and actual studies on human than actually stuff that was based on general research papers.
That was really awesome. Thank you. It filled me with questions. Probably more than I asked. Well, one question I'm wondering about is, you say that microbes vary from place to place, and they vary over our lifespan and age. But does it vary within an age and in a place from generation to generation? Does anyone suggest that?
Yeah, so even in the same place, your microbiome will change as you age. So at birth,
I'm saying at a fixed age.
oh,
If you look at the microbiome of 20-year-olds
yeah,
in Philadelphia from 50 years ago and compare that to the microbiome of 20-year-olds in Philadelphia today.
well, I don't know about,
Same or different? Does anyone look at?
I don't know about, I don't, so there is this one study that I remember, I don't remember the results, that tracked babies, I think in Sweden for over 20 years. So that was one of the few reports across lifespan, but between generations, I'm not sure. I don't think that is a study that look at generations. I would imagine our society changes, so probably your microbiome will respond to those changes. Now, 50 years, I don't know how big of a change that would be, but if you look even older than that, for sure, that will have changed.
And speaking of looking at babies and their microbiomes, there's a practice in some places in Europe and even historically in the former Soviet Union where they would want to inoculate babies when they were born with microbes. I think the Soviets did it through yogurt treatment of kids when they were toddlers, not babies. But in Finland today, there's a practice of as soon as they come out of the womb, you inoculate them.
Yeah.
And we're not even remotely close to practicing that here. Do you think that's something we should be doing?
Well, the effect of C-section on babies. So like in my old lab, people did a lot of C-section work, and whenever people did posters, didn't do C-section work, but a lot of people do like posters, and like a mom who see that was in a different field, and they would talk to the researchers like, "Oh, worried, will my baby be okay later in life because they were born by C-section?" I was born by C-section. I'm okay. So the thing with that, those findings is that what we see is that when the babies start to grow, the microbiome kind of normalizes. And I think that most cases it's okay, but I don't. What research is also showing is that having that initial seeding by collecting the mother's vaginal slash anal microbiota and inoculating that to the baby, that actually helps in the first, in the beginning of life. So I'm not a doctor. I'm not sure how helpful that it actually is in terms of clinical aspects, but it's something interesting that people are moving towards. Yeah.
And another one, I'll keep going until we're at. Um, a pet subject of mine, as indicated in your picture there, from
Yeah.
the person there eating yogurt, is that there is a certain understanding that a lot of the benefit we get from microbes fermenting in our gut, in our colon, is what they produce. Their metabolites or output end up in our blood. So like 1% of our blood is vinegar right now as we're all sitting here standing. There's enormous quantities of protein fatty acids in our blood. And that all comes from absorption from our microbes we produce. We can get a lot of the same things by eating fermented foods. Do you know of any evidence that the microbial output generated ex vivo that's absorbed through the gut does similar things to what's produced in the gut and absorbed?
Sorry, can you repeat?
So we can either make metabolites from microbes in our gut and absorb them, or we can make them out of our body
and eat them.
Fermented foods. I don't know of any evidence that they do the same thing.
Well, I'm not sure. I'm not aware of a study that compared that. I know the studies that compare the people intaking high levels of fermented food versus people intaking low levels of fermented food. And in general, the people who eat higher fermented foods tend to perform better in cognitive tests and seems to be a little bit more resilient to stress. So that's what I know. If we can replenish but the microbes by doing that, I don't know. But also when you're eating the fermented foods, you're already introducing some of these microbes to your community as well. So I kind of think that that becomes a confound where you cannot really understand what is the effect of them separate by the ones that you're actually producing in your gut.
One last question.
Yeah.
This is a Monell question. So we know that taste receptors and olfactory receptors are regulators of immune responses. And we also know that smell and taste receptors, especially taste receptors, are expressed throughout the GI tract from A to Z all the way through it. And there's an idea that I don't know how much evidence there's for, but there's an idea that the taste and smell receptors in the gut, as they regulate, actually control the microbiome, help shape it at a say a genetic level. Individual level. So my question is, are you going to study that? And if not, why not?
So my focus would be in metabolism and food intake and trying to understand how the microbiome shapes food intake. So I'm more interested in how microbes shape the brain and how we eat, and not trying to understand how the microbiome itself is shaped. So unfortunately, no, I'm not going to be studying that, but I'll keep that in mind. So like immune cells that are resident to the gut, they are known to be very important regulators of the microbiome. And this is what is really important to remember, that a lot of people focus on how the microbiome can shape the brain and shape all these aspects of health, but we are ourselves shaping our microbiome. I'm not super familiar with the taste receptor literature in the gut to know how they affect the immune cells itself, but yeah, like immune cells in the gut produce a lot of antimicrobial peptides that will shape your microbiome and control the microbial population.